All right. Hello, everyone. I'm Chloe Halfhide, and I'm the Scientific Application Specialist at Precisionary Instruments, and welcome to today's webinar. Our speaker today is Dr. Carlos Manlio Diaz Garcia, and he has an impressive background in neuroscience. He earned his undergraduate degree from the University of La Habana in Cuba, where he researched autonomic dysregulation in patients with spinocerebellar ataxia type 2. His master's degree is focused on zinc modulation of calcium channels in rat ventricular cardiomyocytes, and his Ph.D. research centered on the TRPV1 channel's role in insulin secretion and glucose tolerance. During his postdoc at Harvard Medical School, Dr. Diaz Garcia studied energy demand and supply in the stimulated brain. He is now an assistant professor at the University of Oklahoma Health Science Center, researching neuronal activity, metabolism, and brain insulin signaling. In today's webinar, Dr. Diaz Garcia will discuss using acute brain slices to explore cellular metabolic responses to neuronal stimulation. He will cover the benefits of this model for visualizing neuronal metabolism using genetically encoded fluorescent biosensors, 2-photon fluorescence, lifetime imaging, and electrophysiological readouts. Additionally, he will talk about advancements in preparing for proteomic and respirometry assays, shedding light on metabolic cooperation between astrocytes and neurons and the regulation of NADH dynamics by calcium-2 fluxes. By the end of this webinar, you will understand the advantages and disadvantages of using acute brain slices for studying neuronal metabolism. Dr. Diaz Garcia will explain methodologies for visualizing neuronal metabolism in live brain tissue and discuss published research using these techniques in acute brain slices. Please join me in welcoming Dr. Carlos Manlio Diaz Garcia.
Well, good morning, everyone. Thank you so much, Chloe, for such a nice introduction. And thank you also to Dr. Xu for the invite to present here today. The title of my presentation today is Investigating Neuronal Metabolism within the Preserved Cellular Environment of Acute Brain Slices. And this presentation has been basically organized in 3 sections. The first one on the neuronal metabolic responses to stimulation. The second one on the rapid calcium regulation of neuronal metabolism. And finally, I will speak about a particular mechanism in which mitochondrial calcium fluxes regulates both neuronal metabolism and, we believe, brain function in the form of cognition. So as a neuroscientist, I have been always fascinated by the many feats that the brain performs at all levels, from the cellular level and to the organ level, right? So from gene expression, excitability, neurotransmission to higher order functions like cognition. But as a biochemist, I have been more interested in the energy required to sustain these functions. And just to give you a perspective, the brain that only represents around 2% of the body weight based on oxygen consumption estimates can consume at rest around 20% of the total energy budget. And that puts the brain apart to highly consuming organs like the whole skeletal muscle at rest, right? But paradoxically, the brain does not have extensive energy reserves in the form of glycogen. And the reserve that it has is basically in astrocytes, away from the neurons, which are supposed to be the ones where most of the action happens, right? That means that the brain relies on the constant supply of fuels, which comes mostly in the form of glucose. And this reliance on the constant supply of fuels, in combination with the high energy demands and the scarce energy reserves, confers some metabolic vulnerabilities to neurons that are manifest when the fuel supply or the oxygen availability gets compromised, like in these conditions, which can lead to a plethora of symptoms and complications that can range from mild fainting, which is a temporary condition, to more severe conditions, including the loss of neuronal stomata and possibly neurodegeneration. But energy demands are not only high in the brain, they are also highly dynamic. And let's illustrate that through an example of how neuronal activity happens. So this is an acute brain slice from the hippocampus. We will talk a little bit more about this further down the presentation. But what I want to illustrate that we can use this to place a recording electrode, a patch clamp electrode, and record the electrical activity of one of these neurons within this brain slice. And you will see that, sorry, this is not playing, I apologize. This is not playing. Oh yeah, so now it's playing. I apologize for that. So basically you can see that while it has some resting membrane potential, so upon stimulation, they can produce these changes in the voltage or the currents associated with these voltage changes across the plasma membrane that are sudden, and it comes in trains, in bursts of these variations in the membrane potential. And this happens through ion channels, mostly sodium and potassium. I have only represented here sodium for simplicity, which then alter the ionic gradient throughout the membrane, which are then restored by the actions of ionic transporters, in this case, the sodium-potassium ATPase, the sodium-potassium pump, which are a big component in the brain. They consume almost half the energy there. And of course, these ions are moved out of in the case of sodium, it's extruded. In the case of potassium, it's restituted back into the cell at the expense of the hydrolysis of ATP, which is the metabolic currency in the cell. Now, the electrical activity of neurons, or the electrical activity of any excitable cell, basically virtually any excitable cell, also involve changes in the calcium concentration intracellularly, which can be visualized with calcium indicators. They can be dyes or they can be proteins that we will discuss further down as well. Here, we're representing the same slice expressing a genetically encoded fluorescent biosensor. And as you may have seen, there are waves in the intensity of this sensor that reports on increases in the calcium concentration. And calcium is not only a signaling ion, but it's also an ion that is it is costly to maintain at low levels at rest because, again, of the action of either ion pumps that extrude the calcium out of the cells, or transporter that exchange the calcium by sodium and then basically pay for this extrusion of calcium through the sodium-potassium pump. So we are very interested in how these events that involve ion fluxes and the energy consumption to restore the ionic gradients after this electrical activity signal down to glycolysis and the mitochondrial metabolism to basically rev up energy metabolism and sustain this electrical activity. Now, this topic has been intriguing because of a puzzling property of the brain tissue, which was described in the 80s by Fox and Rakely, and that is that at rest, all the glucose that is broken down to pyruvate appears to be well coupled to the pyruvate oxidation in the mitochondria, right? But when the brain is stimulated, and in this case, the authors use visual stimulation in human subjects and follow several metabolic parameters through positron emission tomography, they found that when they visually stimulate the brain of these individuals, there was a 50% increase in the blood flow in the visual cortex, which makes sense. That would provide both nutrients and oxygen to the stimulated area. And the glucose uptake in that area basically mirrored this increase in availability, right? It's almost a 50%. But interestingly, the increase over the baseline of the oxygen consumption rate was several fold less than the increase in glucose, which suggests a mismatch between the ability to break down glucose through glycolysis and the ability of mitochondria to uptake pyruvate. And that suggests that a fraction of this excess in pyruvate gets converted to lactate through the action of the lactate dehydrogenase in order to pre-oxidize NADA and make it available for glycolysis to run. In other words, that the brain parenchyma becomes lactate exported, at least transiently, after stimulation. And this process has been named aerobic glycolysis because it's basically a fermentation that happens in the presence of enough oxygen to run it into mitochondria otherwise. And that has been very intriguing, why this happens in the first place. But even more controversial has been where this happens, or basically the cellular loci where this happens. And there have been two competing hypotheses to explain this phenomenon. involving neurons, for example. One in which synaptic activity triggers glucose utilization within the neuron themselves, and likely in other cells, in neural cells as well. But since the 90s, since the last decade of since the 90s, there has been an emergence of a very popular hypothesis in which proposes a special segregation of these functions, in which glutamate release from synaptic terminals, as well as glutamate efflux during the firing of action potentials in the postsynaptic compartments, triggers glycolysis preferentially in astrocytes, which then overproduce lactate, which is transported out of the astrocyte through monocarboxylate transporters, and then into the neurons through similar transporters as well. And then neurons becomes preferentially lactate importers and consumers during stimulation. So we realized that in both cases, in the two proposed hypotheses, there should be an increase in the NADH NAD plus ratio, because glycolysis involves the glyceraldehyde phosphate dehydrogenase enzyme, which basically reduces NAD plus to NADH, and the equilibrium between lactate and pyruvate through LDH, it's basically based on the same redox couple. So we wondered if we can measure this ratio in live brain tissue, and whether we can harness these measurements to basically test this hypothesis. So how can we do that? Well, a lot of the results that I will present today were actually performed during my postdoctoral training in the laboratory of Dr. Gary Yellen at Harvard Medical School. And his group had previously produced a genetically encoded fluorescent biosensor that actually measured the NADH and NAD plus ratio. It combined both molecules, so basically they compete and they change the ability of this protein to fluoresce. When they bind NADH, the protein emits more photons. When it bounds NAD+, it becomes dimmer. But not only that, this fluorescent biosensor also has a fluorescent lifetime change. So what do we mean by fluorescent lifetime change? So fluorescent lifetime is the time that the chromophore spends in the excited state before returning to the ground state by emitting a photon, which is the process of fluorescence. And we can measure that by providing a laser pulse and then following the emission of photons over time. But this happens in nanoseconds, in a span of a dozen of nanoseconds. And then, as you can see here, the characteristic decay, it's exponential. So we can fit this decay to an exponential and calculate the characteristic time, which is representative of the average lifetime of the molecules in the population. And more importantly, for example, when it binds to certain analytes, or the lack of binding to others, can make the chromophore within the protein more exposed to collisions with the solvent, in which case this relaxation to the ground state can occur through a process of quenching, which is non-radiative. So basically, these collisions quenches the ability to fluoresce, and therefore provide an avenue that will not produce a signal, or basically attenuate the signal and speed up its decay. So we can follow the change in this fluorescence lifetime in these exponential decays as an indication of the binding of NADH and NAD+ into the molecule. And this is very advantageous because of it becomes these measurements becomes independent of sensor expression levels. So sometimes we cannot control how much of the plasmid gets expressed in the cells, and that may produce a variability in the intensity levels that can be misinterpreted as changes in the analyte levels. But in this case, the fluorescent lifetime will be similar, independent of expression levels, if the analyte concentration is similar among cells. It is also intrinsically quantitative, so we can calibrate in vitro or in permeabilized cells the values of this fluorescent lifetime to known values of the analyte concentration. And then we can basically interpolate the measurements that we perform in vivo or in ex vivo. And by using this single single fluorophore protein, because this is a single fluorophore, we can harness other channels within the microscope and then multiplex by color. Unfortunately, we were able to do so using this fluorescent biosensor that is sensitive to calcium. Its name is Arkham. It is a family of sensor actually, but it has a prominent fluorescent lifetime change as well. So this fluoresces in the green, this fluoresces in the red, so they are spectrally compatible with most of the common filters used for microscopes. Now, how do we get these sensors to be expressed in the tissue of interest? So we focus on the hippocampus, the mouse hippocampus, and we basically package the genetic information for these biosensors into adeno-associated viruses that we then inject into the mouse hippocampus. And after 2-3 weeks, we obtain acute brain slices using Precisionary instruments. And I want to emphasize that we have adopted these instrumentation and these protocols for several reasons. One of them is very user-friendly, and it's very easy to train new lab members, including the more junior ones, and they quickly can learn how to use the equipment and successfully obtain viable slices. It also produces very reproducible cuts, so that's very important. Especially if we need down the road, I will talk a little bit about that. If we need precise and reproducible sizes or weights for certain assays. And then we have also observed that overall it preserves very well the health of the tissue. So we have been able, there's no problem, to perform experiments to a max of 3 to 5 hours. But in some cases, we have been doing tests beyond that time, and the slices perform pretty well. So it can give you a lot of flexibility in the experiment that you perform. And we harness the advantages that the acute brain slices provide, which are, for example, minimal cellular remodeling when compared, for example, with culture of neuronal culture, primary cultures, or organotypic slices that it is well known that some of the cells die, and they also reprogram their metabolism very quickly, so may not represent, may deviate from how they behave in vivo. Also, because there are compact layers of neuronal somata that we can image with confidence that we will have several neurons represented per field of view, and also very well-defined pre- and postsynaptic compartments. So we know, for example, that in this layer, we focus on the dentate gyrus, which is this region of the hippocampus, and specifically in the dentate granule cells of the dentate gyrus. But we know, for example, that the dendrites project into the molecular layer and the axons into the hilus to then form a bundle of the MOSIF, named the MOSIF fiber tract that projects into the CA3 region. So the circuitry, it's very well defined. Most importantly, for the kind of question that we're trying to tackle, which is a potential metabolic cooperation between two different neural cell types, this preparation preserves or largely preserves the cellular architecture of the tissue, so we can then explore their potential interactions as well. And again, another of the advantages of having such a well-characterized anatomy and connectivity is that we can place electrodes to deliver different modalities of stimulation, like, for example, synaptic stimulation, and we can also place recording electrodes to have an orthogonal measurements of neuronal activity. I mentioned that we measure calcium. We can also measure the electrical activity of the neurons in the form of local field potentials or more sophisticated electrophysiological readouts. And again, like we can move the stimulation directly into the axons. We can do, it provides a lot of flexibility. In addition to that, by having an ex vivo preparation, we have also a good control of the extracellular solution, and therefore we can perform pharmacological interventions or even solution exchange. So it provides a lot of flexibility, although it is an ex vivo preparation. So what do we get when we use this and we place an electrode in the molecular layer to deliver synaptic stimulation? So here you will see a film strip of the Arkham signal, which the intensity has been used for contrast, but it is pseudo-color actually, according to their lifetime. And just So you know the warmer colors indicate an increase in metabolite levels. And you'll see that upon the delivery of a pulse, actually a train of electrical pulses into the molecular layer, it triggers the activation of the neurons, which is visualized as a transient increase in the outcome lifetime. And then it's very fast. It's a very transient and brief increase that resumes very quickly. And you can see the quantitation of these measurements right here with obviously a higher frequency, right? But what happened to the pedagogues sensor? So we observe indeed a delayed but sustained increase in the NADH levels in the cytosol of these cells. And you can see here, they actually outlast the stimulation, which was very interesting for us. So we wonder if this was only a manifestation of a very strong stimulation. So where this could be actually physiological. And to do that, we vary the intensity and the number of pulses of the stimulation to have a range of strengths of stimulation into these neurons. And we then plot the magnitude of the pedagogue response in the y-axis versus the magnitude of the calcium response, which is a proxy for the level of neuronal activity. And what we found is that they were tightly correlated over the whole range of over the whole dynamic range of the sensor, the calcium sensor that we can measure activity, which suggests to us that this is not just an artifact of an excessive stimulation, but rather a greater response to the level of activity within the cell. Now that we know that we are able to assess that in live brain tissue, in neurons specifically, we wanted to design an approach to test the hypothesis that we talked about in the beginning. So we started with whether glutamate is sufficient. So the glutamate release from synaptic terminals is sufficient to engage the associated glycolysis and therefore the lactate transfer into neurons. And to do that, we obtain the responses at baseline and then watch in a cocktail of synaptic blockers. Well, specifically the ionotropic glutamate receptors, which mediates the excitation in the postsynapses. And that, as expected, blocked any calcium response in the neurons of interest. But surprisingly, it didn't produce any increase in the NADH and NAD+ level that would be expected from lactate being transferred into the neurons. So basically, with the preserved potential glycolytic response in the astrocyte, we couldn't see any increase in the NADH and NAD+ level reflecting some lactate import into neurons. That was almost complete. And just to corroborate that these neurons were not dying or anything compromising their health, we removed this cocktail, and we, as we removed the drugs, we repeated the stimulation, and of course we were able to recover the NADH responses. So that prompted us to conclude that the astrocyte stimulation by glutamate is not sufficient. But what if the stimulation of the postsynaptic compartments of the neurons of interest is actually necessary? Because glutamate is not the only trigger for astrocyte glycolysis. The potassium efflux during the action potentials of these neurons has been also proposed to be a trigger for that. So we decided to design a different experiment in which we preserve all the stimulation into these neurons, but then disrupt the potential transfer of lactate from astrocyte to neurons. And to do that, we obtain, again, our responses in control conditions. We built our relationship between the magnitude of this response versus the magnitude of the calcium spike or the neuronal activity. And we then assess whether indeed by changing the lactate concentration in the extracellular space, we were able to produce a response in neurons. So we exposed briefly the cells to a high concentration of lactate without any kind of stimulation. And yes, we were able to see a transient elevation of the NADH level in the cytosol of the neurons. And then we watch in an inhibitor of the monocarboxylate transporters. And this will inhibit mostly the neuronal, but part of the associated as well. And we then observe that these significantly reduce the ability of neuron to uptake the lactate when exposed in the cellular solution, which gives you a quantitative measurements of how much of this blockade happens in this neuron, which is almost 70%. So if the astrocyte to neuron shuttle was true, we would expect that during stimulation, these transients would become attenuated by the same factor, and therefore they would be distributed along this line. However, when we perform an stimulation in the presence of this inhibitor, what we observed was not only the transient was preserved, it became bigger. And you can see here as they distributed in a higher slope. And what does that mean? So we believe that what that means is by preventing lactate fluxes in the neuron, lactate gets accumulated, and that clamps the NADH and NAD plus ratio, at least transiently, to a higher value, which means that during stimulation, it is unlikely that neurons become lactate importers, but rather lactate exporters, because clearly this NADH increase is now coming from the outside. So we thought, well, what if we remove this engagement of the astrocyte by preventing any signaling or any release of glutamate in our preparations, even preserving neuronal activity and therefore potassium influx. So to do that, we basically move the stimulating electrode to the axons of the cells, and we deliver the stimulation into the axon to see if we could obtain any response at all. And surprisingly, we will obtain very similar responses that even overlap with this relationship between the magnitude of the heterox versus the magnitude of the ARCAM sensors. So basically, this is indicating that glutamate spillover near astrocytes is not even necessary for obtaining these metabolic responses in the cytosol. Now, of course, that will lead us to this idea that the neurons themselves are becoming highly glycolytic during stimulation, or more glycolytic during stimulation, and even lactate producers. So in order to test this hypothesis, we use a biosensor, another genetically encoded fluorescent biosensors, from the group of Dr. Felipe Barros in Chile. And we deploy it in our neurons, and we perform similar synaptic stimulation. Again, we were able to detect transient increases that basically match, for the most part, the time course of the transient that we saw for the NADH and NAD plus levels. And we repeated again the inhibition of the monocarboxylate transporters and challenge these neurons with extra stimulation. And again, we observed that these transients did not go away, but they remain basically unaltered. If anything, slightly, they trend slightly to higher values. So again, this is highly consistent with our conclusions that lactate is being produced inside the neuron, not being uptaken. Now, can we determine whether it is coming from glycolysis? So that is the question. Where is this coming from? Well, the most likely explanation is from glycolysis, but we need evidence for that. So in order to do that, we designed an experiment in which we obtain the responses, the initial responses. We then provide lactate at physiological level, like 2 millimolar, in which they obviously, some of this lactate will be taken up into the neuron, and it will elevate the baseline, of course, because it will move some of this into this direction, some of this reaction. But upon stimulation, that didn't prevent these transients to occur. So what we did next is, like, even in the presence of lactate that should supply enough intermediates into this pathway, we're going to block glycolysis using an inhibitor of the GAPDH enzyme, which we found to be pretty challenging because it really compromised the health of the slices. But we managed to find conditions, and when we went to room temperature, just apply it for just cause a partial inhibition of this enzyme, and then return to near physiologic temperature and repeat the stimulation. We observe massive calcium spikes, which speaks of deregulated ability to buffer this calcium or to extrude this calcium. But more importantly, we observed a severely attenuated paradox response, which again supports the idea that this increase in NADH it's mediated by the action of the gap DH enzyme that participates in glycolysis, even in the presence of enough lactate to be utilized. So that is another, I think that is one of the strongest evidence that neurons are indeed capable of this aerobic glycolysis. So how much of this is actually, so we can conclude that definitely glycolysis contributes to this response, but how much of this elevation of the NADH levels correspond to an increase in glycolytic flux versus, for example, a decrease in the ability of mitochondria to clear some of this NADH. And I say this because there are, NADH is, for the most part, impermeable into the mitochondria, right? But there is a system, and typically pyruvate gets into the mitochondria and gets utilized by the TCA cycle. And then there is a system of proteins and enzymes and transporters that can mediate the oxidation of NADH to NAD plus in the cytosol at the expense of the reduction of NAD plus to NADH in the mitochondria. So the system functions at the expense of the proton gradient of the mitochondria. So an energized mitochondria is required, but basically it does the same job than LDH in terms of reoxidizing NADH back to NAD plus. So we consider the possibility that this increase in the NADH in the cytosol upon stimulation may rely on the production of glucose to pyruvate, but basically it could also be in part due to a slowdown of this reoxidizing mechanism that would basically increase transiently the NADH as well, forcing pyruvate to go into the LDH reaction. So in order to test this, we decided to block the malate aspartate shuttle. And indeed, when we did this, the paradox signal went almost to saturation, which means that in neurons, this system is prominent, which agrees with the work of Dr. McKenna and others. So this is known that it is an expected result. Now, the question is whether this is the mechanism that actually produced the NADH transit. Now, we have two things. We have several caveats to continue just with this approach, because for one, the sensor is almost saturated, so that prevents our ability to detect any change further up of these values. And also because inhibiting the malate aspartate shuttle is also known to basically force pyruvate to form lactate because of the increase in NADH levels, which can also deprive the mitochondria from pyruvate. So that is a confounding factor that we have to take into account. So in order to deal with these two caveats, we provide extra pyruvate, like 1.5 millimolar, which would provide enough pyruvate to feed the mitochondria, but also some of pyruvate to clear up this excess in cytosolic NADH and therefore lower the baseline to the paradox baseline to values near the physiological initial values. So now we can test variations in the paradox signal in this range. So of course we have to obtain our controls now in the presence of pyruvate in the solution. But even in the presence of pyruvate, which also feeds the mitochondria, we still obtain clear paradox transients in the cytosol. And when we apply pyruvate in addition to the malate aspartate shuttle inhibitor, we again obtain pretty much the same responses, which makes us conclude that these transients are actually due to an increased glycolysis rather than any slowdown of the malate aspartate shuttle. So all our evidence is so far consistent with higher glucose utilization. But can we measure this? And the answer is, like, we work with some of our collaborators, the Luger lab in Janelia, to obtain a glucose-sensitive biosensor that also present a lifetime change. And as you can see here, when we deliver stimulation, we again reveal transient decreases, so basically these dips in here indicate a decrease in the glucose concentration, which is consistent with higher glucose utilization. So when we apply an inhibitor of glucose transport, again, the baseline went down, and we deliver the stimulate again, the transients became deeper, indicating that in the face of limited supply, increased energy demands leads to definitely a utilization of glucose inside the cell, or at least this is consistent with that. Obviously, this has some caveats, this pharmacology, so we had to perform some controls with the related compound that can also affect the cytoskeleton, but as you can see here, this compound, which is cytochalasin D, did not affect the glucose readouts, whereas cytochalasin B makes these dips even bigger, as one would expect of glucose utilization. So taken together, we believe that our data is highly supportive of direct glucose utilization and performing aerobic glycolysis in stimulated conditions rather than the use of external lactate to support the activity. We also wanted to understand what happened in let's say with the respiration of the cell, because we have a bona fide measurement, we believe now, of glycolysis, but we want to know if we can assess mitochondrial metabolism with a readout, with a similar readout. Unfortunately, our sensor would become saturated in the mitochondria, so we cannot. We can target it, but it will be saturated, and we will not have the ability to detect any changes in here. So what we did was, like, well, first let's focus on this relationship of activity and calcium signals, and create paradigms that we can then test on the mitochondrial function. So first of all, we said, okay, so let's try to see what calcium does. We know that calcium is expensive to extrude, so if we take that out, we would expect this signal to become reduced and probably get away completely with it if calcium was the main ion. When we did that and we measured the pedagogue signal, we observed a significant reduction of this transient, but not a complete elimination, which suggests that sodium alone, without the presence of calcium, still produce some of this signal. So we rationalized that that if we can increase these fluxes, we can take the energy demands to a level that could mimic the energy demands associated with calcium extrusion. And to do that, we use this neurotoxin, which is alpha-pompilidotoxin. It prevents sodium channel inactivation, so it broadens the action potential, basically, and the influx of sodium, therefore revving up the activity of the sodium-potassium pump. And when we did that, we were able to basically titrate to a point that we could restore most of the original magnitude of the response. So again, this is indicative that calcium is not strictly necessary for the response, but rather the energy demands associated with the extrusion. And subsequent work in the Jelen lab by Dylan Mayer and colleagues show that indeed it is the sodium pump, the one that dominates the regulation of glycolysis, but that calcium is a major contributor to these energy demands through the exchange with sodium. So basically, most of the calcium gets exchanged by sodium, and then sodium activates the sodium-potassium pump, leading to this response. So that solved that mystery. Now, again, like, can we harness this knowledge to kind of explore what's going on in the mitochondria? So again, we were not able to use this sensor, but we harness the autofluorescence of brain tissue, which has been shown to the UV-excited autofluorescence of brain tissue, which have been shown to be to reflect changes in mitochondrial NADH pools for the most part. And just to give you an idea, this is so we lose some of the cellular resolution by giving this precise anatomy. We can confidently draw regions of interest on the neuronal somata and measure their responses there. And basically, you will see here that upon stimulation, the intensity goes transiently down, but then there is an increase in intensity following that last several seconds or sometimes minutes, depending on the strength of the stimulation. So this is a stereotypical biphasic response that we obtain using UV-excited NADPH autofluorescence. And we couldn't avoid noticing that this NADP, so I want to make a distinction. It is called NADPH because NADH and NADPH are spectrally undistinguishable. But for the most part, several pharmacological measurements report that NADH basically is the one that dominates this signal. So I will interpret them on the light of NADH, even though I cannot certainly, nobody can certainly distinguish based on the spectral properties whether it is NADH or NADPH. But this biphasic response involves an initial dip, which reflects NADH oxidation, followed by a buildup in NADH or reductive phase that we will call overshoot. And these two overlap a lot. So again, it came back to the idea of, is there any crosstalk between these two signals? So to put that question to rest, we designed an experiment in which we sought to inhibit one of the transients while keeping the other intact. And one idea that we had is, like, what if we block pyruvate supply into mitochondria? What happened here and what happened in the cytosol? So in order to do that, we had to also block the lactate dehydrogenase, because we found that if you block the pyruvate uptake into the mitochondria, it will accumulate pyruvate in the cytosol, which will then force this reaction back into lactate, and then it will basically force a reoxidation of cytosolic NADH into NAD+, lowering also the NADH NAD+ ratio in the cytosol. So we want to preserve it for the most part. So we block the LDH reaction, which is kind of similar to the MCT experiments. So basically, these would, in principle, increase the NADH NAD+ ratio in control conditions, which it does. And then in these conditions, we block the mitochondrial pyruvate carrier. And we had the same recordings in the mitochondria where the inhibitor of LDH basically increased the baseline of the NADPH signal in the mitochondria, indicating that at rest, when we block the ability of pyruvate to go into lactate and therefore oxidizing NADH to NAD+, there is an extra NADH that gets then taken into a mitochondria, well, of reducing equivalents that are taken into a mitochondria. But upon stimulation, that doesn't affect the response. But when we apply the MPC, the mitochondrial pyruvate carrier inhibitor, we observe an intact glycolytic response, but a severely attenuated mitochondrial response. So we are confident that these transients are independent from each other, except, of course, when we mess up with common intermediates and do not account for the potential confounding factors. Now, we have this question where, okay, we know that we can get a glycolytic response without a mitochondrial response, but just we wanted to corroborate whether we can have the opposite, like a mitochondrial response without a glycolytic response, or with a highly attenuated glycolytic response. So we'll recall this experiment that we performed with the GAPDH inhibitor, which significantly attenuated the peptide signals in the cytosol. And we compared it to similar conditions in which instead of lactate, they supply pyruvate, but the idea is the same. They feed the mitochondria, and then they inhibited the GAPDH to measure the overshoot in the mitochondria. And they observed that in the presence of mitochondrial fuels, inhibition of glycolysis does not affect the transients in the mitochondria. Whereas we showed that even in the presence of mitochondrial fuels, inhibition of glycolysis definitely affect the ability to produce an NADH in the cytosol. So I think these are complementary experiments that very well support our conclusion that these events are differentially regulated. Now, or at least they occur independently from each other. The question would be, are they similarly regulated? And in order to do that, we repeat the experiment by removing calcium and see what happened to the mitochondrial responses. We observed that the initial dip was largely preserved, but the most sensitive component of this response, which is the overshoot, was completely abrogated. And that resembles our experiments using pompidiotoxin. So we perform application of pompilio toxin to increase energy demands through sodium pumping to see if this ADP production would signal into or will restore the ability to overproduce NADH in the mitochondria. However, even when we double or more the concentration of pompilio toxin, we couldn't observe a significant recovery of the overshoots in the mitochondrial responses, which means that energy demands is not the main driver or the main trigger of the TCA cycle of the mitochondrial metabolism. If the TCA cycle or the overproduction of NADPH, all right, the overproduction of NADPH. So we wonder, well, what could be the trigger? So clearly they are sensitive to calcium. So we decided to block calcium entry into the mitochondria by genetically downregulating the mitochondrial calcium unipolar, which is the major calcium entry route into the neuronal mitochondria. And we did so and then express our calcium sensor into the mitochondria to check whether we had a functional downregulation of the mitochondrial calcium dynamics. And that was indeed the case. And when we test what happened to the NADPH signals in the presence of a downregulated mitochondrial calcium influx, we basically mimic what we observed with UK 5099, which is a reduction in the overshoot, a significant reduction of the overshoot. Now, this is consistent with previous reports that mitochondrial calcium can increase the activity of several dehydrogenases in the TCA cycle. And this result is not particular to the neurons that we were studying, because recent studies from the Mavicar lab has shown that both in hippocampal and cortical neurons, this is also the case when they use a pharmacological approach to inhibit each other. So that's provides some consistency in our findings. Now, what is causing this is only because we have to consider that of course calcium have direct actions, but disrupting calcium dynamics can have other consequences in the cell because of the pleiotropy of calcium atoms. And we sought for changes, let's say, in expression levels of proteins. And we harnessed the same slices, but now we made further microdissection of these slices to isolate the dentate gyros and therefore enrich our samples with the cells of interest. And we were able to detect that in addition to this rapid control, we observe an inhibition of or a reduction in the enzymes that produce pyruvate in the first place. So interesting, these cells lack basically pyruvate production and pyruvate utilization. How do they overcome that? So we measure, in addition to NADPH, autofluorescent FAD, which will provide what's happening at the level of the succinate dehydrogenase, and they should mirror each other. And indeed, they were attenuated, but they were less attenuated than the NADPH responses, which suggests that these cells may basically present some metabolic flexibility in which they can use probably glutamine or glutamate and run through the TCA cycle to produce some energy through that. They can also produce, or they can also harness more of the NADH producing the cytosol through the shuttles into the mitochondria. And that has been recently reported by the group, the Sumeyer group in dopaminergic neurons, where inhibiting this in the downregulated MCU neurons compromise the production of ATP. So we sought for some evidence of that using cytosolic NADH and NAD plus ratio, and we observed significantly reduced NADH, NAD plus increases during stimulation, even when accounting for the lactate dehydrogenase enzyme, as you can see here, which it's not definitive, but it is consistent with this idea that there is also higher shuttling from the cytosol to the mitochondria. Of course, whether this is because of rapid regulation or involve any change in expression is a question of these shuttles. It's a question to be addressed in the future. So we also wanted to understand ultimately how that affects oxygen consumption. And we take the same slices, these micro-dissected slices, and perform some assessments of the mitochondrial proteins, both the TCA cycle and the electron transport chain. This is by Western blots. We couldn't see any difference. We did targeted proteomics in collaboration with Dr. Kinter, the immunoassay with Dr. Galvan here at UHSC and OMRF. And we couldn't see any change in the protein levels. So that means that the metabolic infrastructure in the mitochondria is intact. Now, we perform how the function works. And we, in collaboration with Dr. Logan and other of our collaborators, we use 2 of these microdissected slices to perform respirometry assays. And upon the addition of different mitochondrial fuels and ADP to promote respiration, we were able to detect oxygen consumption. And that oxygen consumption was actually preserved. It's not trending to higher values in the downregulated model of the MCU. So basically, the metabolic infrastructure to perform respiration is there. But as we have seen later, previously, fuel supply and utilization may be compromised, at least from pyruvate. So how does that play out in the tissue itself? So to assess that, we use oxygen electrodes and perform the same kind of stimulation to assess how oxygen consumption may have been affected by this downregulation in NADPH or NADPH signals. But we observe definitely certain marginal decrease and probably a delayed peak on the oxygen consumption, but it wasn't that. It was the downregulation. It was not as severe as what we observed in the NADPH ratio, which makes us think about 2 things: whether this oxygen consumption reflects mostly ATP production or reactive oxygen species, and whether these signals reflect mostly NADH or NADPH ratios. That is a question that we would like to address in the future. In the meantime, we assess the capacity of this tissue to produce hydrogen peroxide, and we couldn't detect any difference in the regular conditions, only when we inhibited the ATC. We are not sure yet if this has any relevance in physiology. We're trying to explore that, but what it is true is that we have been able to detect through targeted proteomics some downregulation of antioxidant defenses. So let's say this trend toward higher values in the production of hydrogen peroxide in combination with lower antioxidant defenses may suggest some vulnerabilities of the neurons and the MCU is downregulated. So in order to assess whether this has an impact for brain function, we use some of these mice that we wait until they were middle-aged mice. That's when metabolism start to get compromised in the brain of mice. And we assess cognition through diagram water maze assay, also in combination with Dr. Logan. And this assay is basically, we leave the mouse in one of the arms and it is covered by water, so they have to find their way, to learn their way into an escape platform. And they have several trials until they learn this task. Then the target arm is changed, so they got confused. They perform worse. They have many errors. They take a long time to reach or find the platform. But the idea is that controls over several trials, they find their way to the platform again, and they learn it very, very well. Whereas what we found is that the reversal learning of the knockout mice with chronic downregulation in the dentate gyrus did not perform as well as the controls. And when we plot the content of MCU versus the path that they follow, longer path means less performance. They actually correlated inversely, which means that the presence of MCU is associated with a better performance in cognition. And likewise, animals with less MCU expressing the dentate gyrus of the hippocampus perform more errors entering to the previous arm. So they were compromised specifically in the reversal learning, in relearning the task with a different arms. So the takeaways that I would like you to get from this talk is that neuronal metabolism is indeed regulated by neuronal activity through ion fluxes and ion pumping, but that neurons are metabolically flexible and they can adjust to the disruption in some of these pathways. And that, as has been shown in other studies, changes in metabolism can have an impact on brain function, not only at the cellular level, but basically at the behavioral level as well. So in summary, I show you today that acute brain hippocampal slices is a very good model to study neuronal metabolism and function, and the relationship between two. That calcium strongly influence both signals in the glycolytic signal and the mitochondrial signal, even though by different mechanisms, and that disrupting the calcium signaling into the mitochondria remodels metabolism, but also impact behavior. Specifically, we have provided evidence of disrupted reversal learning. So with no further ado, I would like to thank people from my lab for their support and their contributions, both current and former, as well as the internal collaborators that have helped me to move my project forward. But I would like to express huge gratitude to my former postdoctoral mentor, Gary Yellen, and the people in his lab who perform instrumental experiments for the success of many of the results that I presented today, and as well as pioneering even new techniques to make this measurement possible. And of course, I want to thank the funding that I have received over the years and that currently support my laboratory. And thank Precisionary for this opportunity, and thank you all for your interest in this talk and for attending to my talk. Thank you so much. I will be happy to address any questions.
Thank you, Dr. Diaz Garcia. I believe we have one person who put a question in the chat. Sundar, would you like to unmute yourself and ask you your question?
Hi, thank you. Thank you so much for a nice presentation.
Thank you.
So I have a question about the what thickness of the acute hippocampal neuron you took for your experiment, and other question is how long cell are survive in the tissue section. Currently, I'm working on the brain section, try to cut 100 micron and 300 micron. And next target is to do the mitochondria membrane potential using live cell. But when I use the 300 micrometer, it's difficult to stand or making mounting like that. And other question is, I need live cell. How can, after dissection of the mice, how long I can wait to do that experiment? Like in your experiment, you show the many metabolic data. I think at that condition, you need all live cell, right?
Mm-hmm. Well, most of them, yes. So you will want most of the cells to be preserved. So let me ask some more questions. So your first question was the thickness of the slice.
Yes.
Correct?
Yeah.
So we work with anything between 275 microns to 400 microns. I know people who have gone beyond that to 450, right? But I can only speak for my experience in the lab. We work from 275 to 400, and the reason that we change the thickness, it's depending on the application, right? So for some cases, 275 it's enough to preserve the connectivity for placing the electrodes. In other cases, you need thicker because of the tracts are longer, and then you have to ensure that they get preserved over the anatomy. Now, one crucial thing about the health of the slide. So we for publications and for actual data sets, we try not to use the slices beyond 5 hours. That's the max that we will use after slicing. And we always have a 35 minutes of recovery that we keep them at near physiologic temperature to let the slices recover after the slicing. Obviously, I would say the thinner the slice, there is a certain compromise, but thicker slices may have this issue with oxygenation, right? So you have to make sure that during the recovery and during your experiments, these slices are very well oxygenated. If they are not, then that will compromise the health of the slice. So we basically keep them bubbled with 95% oxygen all the time. During the recovery and during the experiment. And that has worked well. Of course, the ones that are the cells that are in the surface of the cuts, they will be most damaged. So expect 25 microns in each side to not be great. But that leaves you with most of the slice in the middle, really in very good conditions. And that if you're using multiphoton, that is super helpful because you can go deeper into the tissue and basically image the cells that are viable. And again, it helps to have, for example, a calcium indicator or some sort of functional indicator that your slices are healthy and responsive.
Okay.
But yeah, no, oxygenation is key. Oxygenation is key. I would also say be attentive to the slicing solution. So pre-oxygenate or oxygenate during the slicing. Make sure that the slicing solution, there are several potential slicing solutions like sucrose base, choline base. So basically preparations that minimize damage during the slicing process. And you may try different slicing solutions to find which one better fits your preparations. That would be another advice.
Yeah, thank you.
I'm sorry, and regarding the loading the cells with dyes, especially brain slices, they are challenging. Typically, and they require larger times. The use of some reagents to help with the permeabilization process that may affect the viability of the cells. So it is challenging. Yeah. So that is one of the advantages of the genetically encoded fluorescent biosensor. You inject them, they express it. You don't have to treat with DMSO or proteomic or CHEM4 things that can deteriorate your sample. But yeah, trying to find the solutions that better preserve the slices and oxygenating them, I think that will help a lot.
Okay, thank you.
No, thank you for your questions.
Thank you. Does anyone else have any other questions? Okay, so if we don't have any questions, I can go ahead, stop the recording, and then we can end the webinar. But if anyone has any questions after this webinar, they can reach out to us and we can get any information necessary from Dr. Diaz Garcia.
No, and feel free to reach out directly if you want as well. I'll be happy to help. Thank you so much again to everyone for their attention, and thank you for the advice.
Thank you. Have a great rest of your day, everyone.